My name is Catherine Colbert, and I'm the director of the Athena Center for Leadership Studies here at Barnard College. And it's so nice to see all of you here. Some of you are familiar faces, others of you are new to our Power Talk series. So welcome, and we're thrilled to have you join us. Tonight we have a great program with Dr. Joan Fallon, who's going to talk about her work and her leadership and her experiences as an entrepreneur. But first I wanted to tell you a little bit about the Athena Center and the work that we are undertaking here at Barnard. The Athena Center started 6 years ago. We have a variety of programs both for Barnard students and for adult women in 3 different arenas. We have a scholars program for students and a whole range of activities that they partake in involving leadership development and programming. We do leadership development programs for corporations and nonprofits, mostly on a customized basis, and this spring we hope to be launching a new executive ed program in collaboration with the Columbia Business School. In the other areas, we work with entrepreneurs in a program we call Entrepreneurs at Athena, and there we do everything from high school boot camps for students to mastermind groups which help young entrepreneurs work together with older entrepreneurs to really solidify their business plans. And most recently, we've undertaken 2 different projects. One is called the Athena Digital Design Agency, which is, for those of you who are around Barnard a lot, we have a tradition of a Barnard babysitting business and the Barnard bartending business. And now we have a digital design business where we teach students to code. They design websites for businesses and nonprofits. They earn some spending money to put themselves through college, and it's a terrific, terrific entry into computer science. So we're thrilled with that program. And lastly, a big effort to push incubators and accelerators to include more women-led startups, and that's called the Athena Pledge. And then lastly, I wanted to invite you, get out your phones now and mark down the dates, February 18 to 21. We sponsor the Athena Film Festival, which is a terrific weekend of features, documentaries, shorts, panels, works in progress, all kinds of glam to celebrate women in leadership in film. Every movie that we show has a woman as the protagonist of the story in a leadership role. It's a fantastic weekend. Please come, bring your friends, bring your daughters, bring your mothers, and it's really an entertaining weekend, so we urge you to mark down that on your calendar and join us then. It is my pleasure to introduce to you tonight Joan Fallon, who is the founder and CEO of Curemark, a biopharmaceutical company that is focused on the development of novel therapies to treat serious diseases for which there are limited treatment options. Of course, nothing but a big, hairy, audacious goal there to find the hardest things to focus on. The company's pipeline includes a phase 3 clinical stage research program for autism and ADHD, as well as 3 preclinical programs focused on Parkinson's disease, schizophrenia, and addiction. So you can see none of these things are easy to deal with, and it's quite an interesting company. In 2013, Curemark commenced the filing of a new drug application for the first novel drug on autism under the FDA's Fast Track program, and that's a designation that's given for the most part only to investigational new drugs that are intended to treat serious or life-threatening conditions and have demonstrated potential. So we're going to hear all about that from Joan. She has dedicated her life's work to championing the health and well-being of children. She holds 46 patents worldwide, has written numerous scholarly articles, lectured extensively across the globe on pediatric developmental problems, including autism and ADHD. She is a former assistant professor at Yeshiva in the Department of Natural Sciences and Mathematics, a graduate of Franklin Marshall. She is a doctor of chiropractic medicine from Palmer University, and she's completed her coursework for a master's in science and clinical investigation from Harvard. And I am thrilled to say that Joan has joined the Athena Center as a distinguished fellow, which is a group of women and men who we honor with being with us for a 2-year term. Jean Sullivan, who just walked in, is another one of our fellows. And we're thrilled to have Jean and Joan as part of our Athena family. So help me give a warm welcome to Joan Fallon.
Thank you.
So hopefully you can see me. These chairs are a little lower than I expected. But let's start. What I'm going to do is start by asking Joan some questions just to have a conversation between us. We're going to come out to the audience pretty quickly to get your take, to get your input into the conversation. I may ask some questions intermittently, but the real goal here is to have an interesting conversation that we can all participate in. So, Joan, let me start by really commenting upon your nonlinear career path. Because I noticed you started at a liberal arts college, then you became a chiropractor, then you have ended up as the CEO of a biopharmaceutical company that, in my view, hopefully will have a breakthrough drug very quickly. So in this non-traditional career path, how did it come to pass, and what got you in the direction you ended up in?
So it's a good question, and those who know me actually started laughing in the audience. So I started out my life as wanting to be a doctor for children. That's something I knew really early on. And I was not exposed to other kinds of healthcare growing up. I just had very traditional healthcare. And went to Franklin and Marshall because at the time they had one of the best acceptance rates in medical school of all the schools. Looking back at the liberal arts education, of course, I'm glad that I did that because of my non-traditional path. But when I got out of school, I applied to medical school, osteopathy school, and chiropractic school as a senior. Got into all three and took a year off and went and visited all the schools that I had gotten into. Everyone thought I was a little bit crazy because everyone's goal was to get into medical school. And so, but I knew that I thought a little bit differently, and I thought about things in a sort of a broader pattern. And so I took the year off, and I went and looked at all the schools, spent a week going to classes at every school that I had gotten into, and was at the chiropractic school, and it was the beginning of the semester. And the professor came in, it was like Diagnosis 101 or something, and very, very basic. And he comes in, it's the first day, and he says, 2 guys go out and play touch football, and one of the wives makes chicken salad sandwiches. And they sit in the car all day long. And they come back at 4 o'clock, they eat the sandwiches, and one guy gets really sick, and the other one doesn't get sick. Which is really the sick guy? And, you know, allopathic medicine would say the guy who got sick. But from a more holistic point of view, it was the person who didn't get sick because his body should have gotten rid of that poison, and it didn't. And I thought, you know what? I think that's for me. So I deferred my medical school another year and went to the chiropractic school. While I was there, I had a tremendous mentor who was both an MD and a chiropractor from New Zealand, who had a wonderful view of healthcare and children, and studied with her, and then came back and never regretted that decision ever. And I don't think if I had gone to medical school, I wouldn't be doing Curemark today.
All right, so jump ahead a little bit and get me to how you got to Curemark and where that came from.
So I think that some of the way that I look at things is from the top down. So if someone is not well or something's going on with someone, it's what's going on with them, what's different or what's changed for them. And so in the late 80s, early 90s, I was seeing a lot of children, and I saw my practice was really children. And they started coming in with a constellation of symptoms I had not seen before. Lack of eye contact, very delayed speech, or difficult speech. And it turns out to be the very early part of what was this autism epidemic. And more children started coming in with these symptoms, and everyone was scurrying and trying to figure out what to do. How do we take care of these kids? Do we give them different medications? Do we do different behaviors? And what, for me, was so prominent that no one else sort of looked at was the fact that while their symptoms were different child to child, so a child may have no speech, some stuttered speech, or they spoke intermittently, what they ate was very much the same child to child. And that really intrigued me. And that sort of led to the beginning of my looking at that and trying to figure out why that would be. Why would their behavioral symptoms be different and sort of what they eat be the same?
So I want to know a little bit more about Curemark, and then we can come back because I have a lot of questions about what you then did next. But tell me a little bit about the company that you founded, and I understand you're really looking at the treatments for autism, which obviously came from that. But tell me more about what you're actually working on now.
So I think, and we'll go back to that, but what we found with the children with autism, and also in some of these other diseases, is that there's a enzyme deficiency. So for example, the enzyme deficiency that we see in children with autism in about a 60 to 70% range, which was the early work that we did, exists in middle to late stage Parkinson's as well. And only in the last week, actually, are they talking about adults with autism get Parkinson's. So there's a definite tie-in there that I recognized very early.
So wait, so you said we, and you said you recognized this early. So tell me how that happened.
So it was really me who started that.
Like most women, it's the royal me. Yes, exactly.
So it was I who saw that, and I did a lot of investigation, and then I got all my buddies, all the pediatricians in town, and everyone else to start testing kids. And so we amassed a data set of over 1,500 children that we actually looked at, and that forms the basis of
Both
Curemark.
those who had the symptoms and those without.
Right, exactly.
And so what made you look at this particular enzyme?
So the children had this very peculiar diet, and I would ask the parents what the children ate, and they would say, oh, they eat the white diet or a tan diet. But when you broke it down, it was carbohydrates. It was, I used to call it the 10-thing diet.
Wait, I'm confused here. So when you said these kids who are exhibiting a variety of symptoms all had the same diet, it was not a typical diet. It was
No, it was not typical. It was,
Okay.
I called it 10 things.
Okay.
And it was French fries and bagels and waffles and chips.
Exactly what I ate when I was pregnant.
Right.
I used to
Right.
call it the white
Right,
food.
right. But that's a temporary state when you're
Right.
pregnant, right? These kids just kept doing that. And that's true of all children as well. They go through these serial, eating type things. But these children did not. They stayed on that theme. And when I asked some of the researchers, I said, what do you think that's about? They said, oh, there's sensory issues, there's this or that. But it was too ingrained, too deep to be just relegated to sensory, because the sensory things for these children differed child to child. They weren't all the same. So that's when I started looking at everything protein. I did blood and urine and stool on these children to see if I can find something that was the same in these children with autism. 9 of them that started this. And they were all had low levels or missing a particular enzyme that digests protein. And that's how it started.
And did that mean they ate the white diet because they didn't have this enzyme and they were trying to counter?
Yeah, I
explain
believe
that for me.
I think it's a self-selection. So if you eat a piece of meat, for example, and you don't have the means to digest it, it's going to leave an undigested piece of meat in your system, which can be uncomfortable and cause other symptoms, allergies and other things. So I think the children had self-selected for these foods. And that, of course, came into the literature much later, that they do self-select for these kinds of foods. And it's pretty profound in terms of
So you started with 9 children
Mm-hmm.
and then developed a database of 1,500 children. And did you find that same pattern in that entire database?
Yeah, we found that between 60 and 70% of children on the autism spectrum had deficiencies in this enzyme.
Okay, so this is like a great murder mystery, right? You gotta get. So what happened next?
Right. So then after the first, we compared those 9 and others to children who had no autism or any other symptoms, and they didn't have that finding. And that finding had only ever been seen in children with cystic fibrosis, and only reported in them. And so we thought, well, maybe we should treat them the same way we treat that deficiency, but that didn't work. It didn't take care of that. And it was only later that I realized it was a different part, the protein piece is what they were missing, and not all of the enzymes.
So what year was this?
That was 1998,
Okay, so a long time ago.
a long time ago.
All right, so fast forward here. How did we get from 1998 discovery of this deficiency to a company that's really targeting this
Well,
problem?
a tremendous lack of sleep. Some phenomenal people who surround me every day, both at the office and out there in the world. And a lot of phenomenal people who have invested in the company and who have believed in this early on. Some of them are in this audience. And because there's no venture capital at all in autism, not then and not now, we had to do a completely different way of fundraising.
All right, so I'm going to get to that question because I think it raises a variety of different issues. But tell me, so the lack of the enzyme then led to a particular drug to counter that, or what happened there?
We had to replace the enzyme, right?
Okay.
And that presented tremendous challenges because that particular enzyme had never really been replaced before. And so it had to be replaced in a certain part in the GI tract. It had to have a certain trajectory as it goes into the body. And so it had to be coated a certain way, delivered a certain way. It had to have the right amount of it. So employee number 1 was that person. And he had drug formulation experience, worked in pharma, is our chief scientific officer now. And he actually found a novel way to coat the enzyme because I had very strict requirements. The parents don't like colorants and fillers and extenders and binders and all the things that you find in every drug, but not in my drug, I told him.
Okay. That's very brave of you.
Yeah. There have been a couple of moments like that.
Okay. And tell me more about why. Because that's basically saying, I'm going to make a drug and to not only solve the problem, but be different than every other kind of drug.
So, people don't start out, or at least I didn't start out saying, I'm going to make a drug, right? It's just sort of each step along the way, I was compelled to go to the next step. I think had I known what would happen between then and now, it would have been more difficult to do. But I didn't know. I just knew that I had to do this. I had to figure it out. I had to say to myself, if this has a chance to help these kids, I have to do it.
So what was the first indication that this actually could help the kids?
Well, I think we did some early work also in trying to figure out what they would need. So we cobbled some things together and looked at a lot of different science around.
Wait, there's got to be more to the story than just cobbling some things together. So help me get the details here.
So we had to replace a particular enzyme, a protease, so one that digests protein. And they don't exist by themselves. So we had to find different things, supplements, other things, prescription medications that have that element in there and figure out how much of that element was needed to make the change in the behaviors of the children. So we did some very early work with some of the local pediatricians to help do that. And we're able to figure out what it was that would change the children and make their behaviors change.
So tell me the story of the first kid who you saw change, or 2 or 3, that what I think of as the aha moment.
Well, I think the real, for me, it's interesting because the drug was not the aha moment. It was the deficiency, the uncovering of that, I call it a biomarker, that really was the aha moment. I remember walking out of my office and looking at my staff and going, I think I've got it. I think that maybe it's an enzyme deficiency. So let's add that to the battery of what we were doing. And to me, that was the aha moment when those test results came back. And we tested them on different days, at different labs, different times of the day to make sure that there wasn't a red herring in there. And every time that computer, you know, the old computer with the pin feed would go off in my office, and those results would come in, it'd be like, oh my goodness, look at this. Another low, another low, another low. That to me was really the aha moment. The drug itself was just a way to sort of fix that.
Now, was there other people in the medical profession working on this theory at the time that you started?
No.
And how about today?
No, people are looking at microbiome is what they're doing. But we've pretty much established.
For those of us who don't have the
Okay.
scientific,
So
give me the details here on what the difference, what's a microbiome versus a
in our
enzyme deficiency.
intestines, we have microbes of all different kinds. Some of them are good microbes, and some of them are not good microbes. And I think that science has figured out that some of these microbes actually can cause disease. And the enzyme deficiency may or may not alter that. But there are more people looking at a gut brain today than they did back in the 90s when I was starting to look at that and really look at it as a potential root cause of a neurological problem. At that time, people thought I was really crazy. You know,
They probably still do, right?
no, not so much anymore.
Okay.
Because all of this microbiome and what happens in your gut affecting neurology, it's everywhere. I did an epigenetics conference I presented last year, and I took my chief scientific officer, and we were like, everyone wants to sit at our table for lunch because we're actually doing something, right? We're actually doing that. But I think it's a very different world we live in today than even 3 years ago. So I think that those connections are much more in the fore than they were before.
All right, so bring me up to speed here on the story of starting the company and trying to get the resources to be able to actually test these enzymes.
So when I left practice, I sent out a letter to all the people in my practice, all the parents, and said, they all know I'm doing this. I said, I have to go and do this. And so I'm leaving practice. And one of them called me on the phone and said, can I make an investment in your company? Well, I didn't really even have a company at that time. It was just something I was going to do. And I thought, yes, if you hold that thought, I'll be back to you. And she and her husband did make an investment in the company. And so we raised money on a convertible note, and then we went to a Series A, and we raised $5 million, which ended up being $7 million, and then we raised money on milestones. Every time we had a milestone and we wanted to get to the next step, we had accomplished what we had set out to do. And
So go ahead.
I promised my investors, the early investors, that I would give them the best clinical trial that was ever done in autism. I couldn't guarantee the outcome, but it would be the best one that was ever done.
Okay, so help us understand the process of clinical trials and then how you get to the best. What criteria are you using when you say it's going to be the best? But first, give us the process, because some of the audience is probably very familiar with this, and people like me are not.
So generally, when one of the things that I talk about is that Curemark really disrupted the drug discovery model. So what we've done is we looked at patients, looked at what was going on for them, figured out what was going on, and then treated that. That's sort of the paradigm. Drug discovery, up until recently, was all about discovering novel molecules that you can patent and retrofitting them into mechanism of action and developing drugs. So they were developed sort of backwards. I look at it backwards because it really wasn't about the patient. It was about what can we patent, what's different, and then retrofit it into a disease. And I think there isn't a better example than all of those drugs that initially came out for Alzheimer's were sort of red herring because it didn't affect really the disease in the same way that they thought that they would. And so because we were using an enzyme, generally you have to do a phase 1, a phase 2, a phase 3, and then a post-approval study. For us, because the enzyme or the matrix of the enzyme, the makeup of it, the basic part of it, has been known to FDA, and we had this body of information that we had gathered about the deficiency, et cetera, they allowed us to go into a phase 3. Immediately. So we went right to a phase 3 because of the
When
safety.
you say it was known to FDA,
because
it's
they use it to treat cystic fibrosis. It's just formulated very, very differently.
Okay.
So cystic fibrosis, the individuals with that have a primary lipase deficiency, so they can't digest fat. The children with autism don't have that. They really have a single enzyme deficiency around digesting protein.
Okay. So you jumped to the head of the line,
Right?
and what did that entail?
Well, that meant having a big trial, right? Usually you do a phase 2, it's smaller, you not have to be so stringent in that trial because it's really more investigational. And so you have to hire a contract research organization from the outside to run the trial. So it's at arm's length from you. You have to develop an entire regulatory infrastructure around that, meet with FDA, submit a protocol, and that's clinical. And then you have to manufacture this drug on an investigational level, and it has to pass a lot of different thresholds in order to be allowed to be put into humans.
All right, so manufacturing a drug, clearly you didn't learn in school.
Right?
So how did you do that?
So my first employee was the formulation guy who did know how to do that.
Okay.
And so after we got through with all of my criteria, we in-licensed a technology that's used in food. And it's basically a oil coating that has nothing else in it, spun in a particular way so that it is actually solid. So the drug really consists of our enzyme and this coating and nothing else.
So when you said and adopted the rules, tell me what you meant by that.
I mean, in terms of
how you wanted this trial to proceed.
So you have it's an interesting, for startup, having to go into very stringent FDA regulatory behavior was a steep learning curve for us, right? But I think we surrounded ourselves with really good regulatory people who've done this over and over and over again, who knew what the rules are, who brought drugs to market, and who are able to advise us about what to do and how to do it. And we developed a very good rapport with FDA. And so we worked with them, and they were able to guide us through the clinical trial.
So have you had results now at this point?
So yes, so our first trial that we did, which included children who were missing the biomarker, we hit our primary and secondary endpoints, and we went back to FDA and said, okay, we hit our primary and secondary endpoints.
So wait, tell us what that means.
That means that you choose an outcome that is what you would like to see. So for us, it was reducing their irritability and increasing their socialization. And we used an instrument, a psychological instrument that's known to FDA, that's well validated, that will hold up against other trials, and we met those endpoints.
So for those of us, I assume that most of the people in the room know children who are autistic, and there's a spectrum of those.
Right.
When you say we met the endpoint, give us some examples of what that would mean for those kids.
So Well, it's a couple things. One is that in decreasing their irritability and increasing their socialization is sort of a monumental task because you're doing two different things, right? You're bringing them out at the same time making sure that their irritability is not so high. So it's sort of counterintuitive those things would happen together. And indeed, the drugs like risperidol, the antipsychotics that are used for autism are used for older children, but also children that have very severe symptoms. And so for our drug, we did it with young children, 3 to 8. So when it's first diagnosed at 3. So believe it or not, we're not able to see those children. We have an arm's length process with our clinical sites and with our CRO. So we can't actually go in and see the kids. We do have a lot of anecdotes that come out. And indeed, there are children who are still in that trial getting our drug. They all get to, after 12 weeks, they all get to opt in to get the drug. Some of them are in there 4 years. And they've done very well.
Okay, so what happens next?
So we went to FDA, and FDA says, we'd like you to look at children who are biomarker negative, who don't have this deficiency, because we actually think what you're doing will help them as well.
Autistic children or all children?
Autistic children.
Okay. And so,
So we have a clinical trial ongoing right now
okay.
at 20 sites, 21 sites around the country, and looking at children, all children with autism.
Okay. And what's the duration of that? How much longer do you have to wait before
We
you'll
should have the results of that, mid next year.
Okay. And then you can apply for the approval of the
Hopefully
drug?
we'll be finished with that will finish our new drug application.
Okay.
We can go forward.
So, and I'm going to come out to you all very quickly, but you mentioned that you had a lot of difficulty raising capital for this venture, and I'm not surprised by that because, of course, in all of the literature, we see a very, very stark difference between women entrepreneurs and male entrepreneurs in terms of their ability to raise capital. Just to give you a sense of this, basically of conventional small business loans, so not bigger ones like you, women only account for about 4.4% of all of the loaned money. So it's very low, and the bigger the company, the worse off women are in terms of getting venture. So tell me how you overcame that.
Well, I think that actually when I was raising money, I wasn't even cognizant of that fact, right? Because I wasn't traditionally...
Ignorance is bliss has really
Well,
far-reaching implications, right?
I'm an athlete, and I was an athlete before Title IX. So I've ignored the gender piece for a very long time in my life. So, but I could not ignore the autism and no venture capital piece. That piece was really hard to ignore.
So when you say meaning compared to other diseases, autism has a much lower rate of investment?
There's no investment. There's no venture capital at all, not when we started and not today, in autism.
And why is that compared to other diseases?
I think because people are, well, first of all, it's new, right? The largeness of the numbers is new. Mm-hmm. People don't know what causes it. It doesn't all look the same. And there's nothing to really hang your hat on. And so when we started, I think there were 6 or 7 drugs in clinical trials, and late-stage clinical trials, and now they've all failed. So I think that the uncertainty around it has been something that people are not willing to bet on.
Okay. So you've said this a couple of times, but I think it bears repeating because it's a good lesson for leadership, which is you keep saying, if I knew what I know now when I'd started, I never would have done this, meaning it would have felt insurmountable. So talk to me a little bit about what is it that drives you and what we can learn from that in taking on a challenge that is, to most people, not one that's even worth taking on.
Well, I think that part of leadership is leading people somewhere. You have to lead them to something. And for me, it was understanding that I had a piece of knowledge, which was that 60 to 70% of these children were missing this enzyme. And I was going to replace it no matter what. And if it helped them, it did. And if it didn't help them, it didn't. But I needed to know if that was something that was going to help them. Because I don't think if I had that knowledge and I didn't do this, I could not live with myself.
So what I'm confused about, I guess, is why other scientists weren't interested in that finding. Is it that they didn't know about it, or they just weren't interested in this particular disease? Why do you think that wasn't?
Well, I don't think they're not interested. I think that they didn't find it, right? And we began a very large patent portfolio way back in '99 from the first one. Okay. So we sort of have that space, and I think that people are interested in it, and they will be interested in it more. But in autism, there's a lot of factions, and so we sort of stayed out of that fray as well.
Okay. So let's go out and take some questions for you. I have a bunch more about leadership and courage and a variety of other things, but I know that there are questions from the audience, so let's go right here. We've got a microphone coming.
Great.
First, let me say I think that your talk was incredibly fascinating. I so enjoyed it. Thank you for sharing all that.
Thank you.
My question is, having gotten this far along, would you be interested in venture capital funding now?
Yes. Because one of the things that you learn is that you don't turn down money, right, as an entrepreneur. Now,
Don't leave it on the table.
don't leave it on the table, right. Although it has to be, one of the things about this as well is that because very little was known, I went back to school to figure out how to do a clinical trial. I knew what the elements needed to be. And if someone was saying, so for example, our CRO said to us right off the bat, you're doing a trial without blood work for children. FDA will never agree with that. And I said, our enzyme is not absorbed in the bloodstream. There's nothing the blood will show anyone. I said, so I don't see a rationale for doing it. Not going to happen, they said. Did it happen? Yeah, it happened. We were able to do it because we did things that way. So being able to have the freedom to operate in that way was really important, especially early on. Now we're, it's a different situation. But yes, we're always interested in talking to people about that. Yeah.
Am I next?
Yeah, you're right next to her.
Okay, I have the mic. Thank you, Dr. Fallon, and congratulations to you and your team on all the success you've achieved so far. We're all rooting for you. I've got a quick question and then a quick follow-up. The first is there anything preventing Curemark from taking CMAT for autism to England? I understand the approval sequences are easier there, and or Europe. And some, as a follow-up to that, so that's question one. Question two, as a follow-up, I have some sources who claim that product itself could be a multi-billion-dollar product. Would you agree or disagree with that? Thank you.
So there's nothing preventing us from doing that, and indeed we're in dialogue with MHRA at present.
Wait, help us
Okay,
with
I'll get there.
the
Yeah.
acronym.
It's the FDA equivalent
Okay.
in the UK. So we are in dialogue with them right now, and so there is nothing that's preventing us from doing that. And the second question again.
$1 billion.
Oh,
Would I be wrong in saying it could be a multi-billion-dollar product?
I think that the answer is yes. But it's not the focus of what we've started this company about. And I think that there's a lot of children with autism. We started, it was 1 in 100, 1 in 88, 1 in 68. It just keeps growing, and it goes across the pond, And it's in China, we've been in dialogue with China, they estimate it could be as high as 1 in 2 or 3 children. So I think that the need for it is great. I think my focus has always been to make sure that as many children who need it will get it, and that's been our focus. So it's much bigger than we thought it would ever be, and never thought that we would be right now the only person standing. There's a great need for other drugs as well, for other therapies. So I don't want to ignore that fact, but I think that our persistence and our desire to get this to the kids is just so important to us and to me.
So let's try it on this side of the room.
Hi. So it sounds like the theory of the enzyme deficiency isn't like bought into by all. Like as I understand it, usually like
Hold the microphone
that
a little
doesn't.
closer.
Is that better? So once the drug is approved, do you foresee that being a problem of actually it being prescribed in patients, especially if you're saying that it works in patients who are not enzyme deficient? Like, does that make sense?
Excellent question, actually. So I think that the testing, right, the deficiency is a deficiency. It's there. It's not like it's a subjective finding. It's an objective finding, right? So replacing it makes a lot of sense. The degree to which children who have autism, who don't have that deficiency, what it will do for them, I don't know. That's why we're doing this trial. And FDA really wants us to do that. And I think whenever the science is, when you have something that's sort of a biomarker, we call it that very loosely, you have to show that the negative doesn't work. And so that's part of the science, which is what we're doing now. And I think that we want everyone to use this responsibly. So we designed the initial trial to just be used by those who we thought it would help. And so now we're looking at all of that. And that will help doctors guide, if it doesn't have efficacy in the children who have normal levels, it will guide physicians to know how to use the drug. And I think that's an important part. And we will have a lot of experience with our enzyme as it's formulated the way it is with children with autism. And we will have tested more children probably than any other clinical program to date. That's a very excellent question, actually.
Thank you for sharing your very, very interesting and inspirational story, first of all.
Thank you.
A question about your thoughts on the etiology of why the incidence of autism is increasing. Is there some thinking that dietary factors that have changed and a change in the types of protein, perhaps, have led to the increased incidence?
You know, I think that anything I say is no different than anything anyone else would say. We don't really know. The thing I do know and I do talk about is that the difference between children 25 or 30 or 40 or 50 years ago, when I was a child, and what they actually receive today and how they're treated, the entire epidemiology of the pediatrics. No, in terms of everything. So, for example, children now, if they have to go to a daycare, for example, they have to be medicated every sniffle. They're exposed to children much earlier. So all of the epidemiology has changed. How we feed children, what we do with them, who sees them, where they go, when they start school, when they start, doing all of those things has changed. So I think that in there lies the answer. I do also think that there is a genetic predisposition. So I think the gene piece is a piece, but it probably will be epigenetic and not directly genetic. I mean, I think in some cases it is, but I think that we'll find that it's probably something turning on and off the genes. And what we know today that we didn't know when we started this was that the amino acids that our enzymes break off the protein actually are involved in major epigenetic issues. So they turn on and off very important genes like the CHOP gene, which does neural pruning. We know children have more neurons. Children with autism have more neurons than they need. And we know that that CHOP gene, which controls the neural pruning, actually is controlled by one of the amino acids that's broken off by our enzyme. So we believe that our enzyme plays not just a digestive role, but actually a very direct neurological role as well.
So let me follow up on the earlier question. Are you hoping that the enzyme has no effect on the broader group of people without the deficiency, or are you hoping the opposite?
I'm not hoping for any. I'm really not hoping for either. What I'm looking at is, so also the amino acids that our enzyme breaks off are very involved with dopamine and serotonin synthesis. They form the basis of those things. And if boosting serotonin in these children, which we know is low, if boosting that helps them, well, that's great. But I don't think I look at it either way. I just want to know who it helps and get it to them as fast as we can without looking at it. Someone from one of the insurance companies, when I said that a long time ago, came up. He came off the stage and he actually came out in the audience and kissed me. He said, because drug companies are always looking to expand their market, and we're trying to make sure that responsibly who needs to get it will get it.
Okay.
How about back here?
Thank you very much, and
Thank
I'm
you.
gonna echo whatever everyone else was saying. Your talk was really informative. I've got a few questions. One, you said that you convinced FDA not to do the required blood work, which is
I didn't convince them. We submitted it without the blood work.
you
and I
submitted it
didn't.
without the blood work. Okay, how do you test then for the presence or absence of the enzyme?
In the stool.
Really? Okay, now. Is it a common test, or are you going to have to?
It's a common test.
It's common, so you don't have to get a biomarker company to partner with in order
No.
to. Okay, that was one thing.
Yeah.
The other is we're talking about
We
the
have
old.
patents on the use of it, however, coupled with our drug and with the disease.
Well, that's
Right.
good.
Yeah.
The other is you were talking about not following the traditional drug discovery model.
Right.
Can you talk a little bit about how the new model, which I will call it, that you're working with, how you're planning to apply that to other areas? Because I know you're interested in other areas. So how would that model work?
Right. So it's patient-centered drug discovery. That's what I've sort of talked about it. All the big pharmas are doing it now, but they're doing it sort of in, what's the right word? With big data. That's how they're trying to do that. Patients Like Me, which is a big website where they gather a lot of data about different diseases. So I think they're looking at it that way. I think the way that we have looked at it is to go to the source. So if you use the physicians that actually see these patients every day, they'll tell you, like, for example, in Parkinson's, we know that protracted constipation of 3 years or more is an early sign of that. Michael J. Fox tweeted it out today. Do you have constipation? Do you have this? Do you have altered sense of taste or smell? These things we know. The physicians knew that a long time ago. If we aggregated that, it would have been better. We could have understood that. So we know that protracted constipation is a sign of, can be a sign of early Parkinson's. Just recently, a study came out where they looked at innervation by using functional MRI, et cetera, of different organs in the body in Parkinson's. The number one was the GI tract. After that was the pancreas, where our enzyme is made. So the science continues to support the premise that we started with. And I think that using those principles for a host of other diseases will actually help to have more drugs discovered and understand what patients need. Margaret Hamburg spoke at Faster Cures a couple of years ago, and everyone on this panel was talking about, yay, we're going to have a genome card in our pocket, and they're going to pull it out, we're going to tell us exactly how to treat this. And Margaret Hamburg, who had the last word, said, that's nice for later on, but we're missing treatments for people who need them today. How are we going to get those if all we do is concentrate on the genome piece? So we need to be looking at what people need today as well.
So, Joan, I noted that you are doing some other trials around Parkinson's, schizophrenia, addiction, ADHD. Tell me how they all.
So we
factor
were
in.
approached by a group here in New York who, addictionologists, psychiatrists who treat this, and said, we think that based upon the enzyme's action on the amino acids, that you can actually help with cocaine addiction. And at first I thought, that's interesting. I can understand replacing the dopamine, which is what is used up in that kind of addiction. But now there's more science. So we know that in cocaine addiction, when you take it once, the brain makes a transcription factor called delta phos B. And that delta phos B promotes the addiction for 6 months. So even if you just take it once, the body's propensity to want more is there. And the phenylalanine, which is why you make dopamine, right, the amino acid that you need to make dopamine, also is what you need to change the results of that delta phos B. So that delta phos B transcription factor allows the neurons to get bigger and bigger and bigger and bigger. The only way they're going to get back to normal and not be craving more dopamine is to give them phenylalanine. And that's what our drug will do. Our drug will break off phenylalanine and maybe make the pool bigger so that these people can draw upon that. It's the opposite of PKU. You know, they do a heel stick for babies, phenylketonuria. The dendrites on the neurons are very small. They're like stubs. In cocaine addiction, they're the opposite. So we're going to start a phase 2 at Albert Einstein and next year, and use functional MRI to look at the brain changes as a result of our drug. And it will have to be formulated in a way specifically for this.
So all of these are the same drug.
Same matrix, but different formulation. Yeah.
Let's go over here.
Hi.
Hi.
I'm curious in the biomarker negative members of your cohort. So did you observe any interesting biochemical trends in those groups of people? Do they fall under patients who are benefiting more from the gut microbiome type research, or is it just, I'm curious about those.
Yeah, we don't know. I don't know. The thing I do know is that when we looked at individuals as they were screened in our trial, so they were screened for autistic disorder, so the very narrow definition of autism, no Asperger's, no PDD in the trial, which is the way FDA would like it. And then we looked at kids who were biomarker positive and negative. The main autism behavioral test to diagnose it was no difference in the children.
Okay.
So it doesn't seem to subset out a phenotype. So we don't really know beyond that.
Yeah. got
So
it.
it's interesting. Remember, autism is a spectrum, so it's a lot like depression. So people are depressed because they have some sort of organic issue that they've inherited. There's bipolar that runs in a family, or someone died in their life and they're sad and they have depression. I think autism is the same way. It's a big spectrum.
Right.
And so not everything is going to look the same. So for example, a child who had a stroke in utero can exhibit autistic symptomatology as much as a child who we don't know what causes it.
Okay.
So it's really not known.
Yeah, thank you. And the other question I have is related to the spectrum. You mentioned that the levels of expression of the protein differ even between the people that are biomarker positive. Do you see any correlation between the severity of autism and higher levels of that decrease?
We don't.
You don't?
We
okay,
don't, no.
interesting. Thanks so
Yeah,
much.
Okay.
Thank you so much for your remarks. I'm curious if you can talk about how you envision the commercialization of the drug, and not maybe so much in terms of pricing, but do you imagine needing to build out, like other biopharmaceutical companies, a large commercial and marketing team? Is that another thing you hope you'll be able to do differently than other small biopharmaceuticals?
Yes. I mean, because we've done everything a little bit differently, I see this as being different too. The autism group is very well segregated. So it's like many of disease, like for example, cystic fibrosis or muscular dystrophy. Most of those children are treated in centers of excellence, and their treatment is pretty much the same child to child, center to center. So there's a protocol, there's a standard of care. In autism, there is none. So we're hoping that this would become the standard of care when a child is first diagnosed, if it's something that will benefit them, depending on what that looks like. So do we need to have a traditional sales force and all the rest of that thing? I don't see that as being necessary. We live in a way, in a life where things are marketed and sold and have access very differently. So I see that. We've looked at a lot of things. We've explored things. I think part of it depends on whether we do it or whether someone licenses it to do. And so we're not really sure what that looks like today. But yes, but we're looking at other ways of doing that. Yeah, it's a good question.
From my political science class, I'm writing a research paper on how differently two countries allocate their resources and funds for research and treatment for chronic diseases that might affect one gender more than the other. Since autism seemingly affects males more than females, so I was wondering if there is bias in the allocation of funds to research on autism or efforts to provide services based on the gender it affects more.
Rachel, let me see if I can. Did you hear that?
I think yes. I think that it's an excellent question, and I think that.
Can you repeat the question?
Yeah. So it's basically, do we see the research and dollars allocated, et cetera? Make sure I have this right. For research in different diseases, allocated based on gender, right? Okay. And I think that. I don't have firsthand experience with that. However, there is a bias against children. Okay, so, one of the questions I was asked when we were talking to one of these bodies outside of the U.S. was, do I really think these children are debilitated? And what I had to remind everyone was that degeneration and debilitation are two different things. So we use debilitation as becoming debilitated. But these children start out life debilitated. So I see that. I do see that there's a lot more, and you probably can tell me better than I can tell you, right? Because that's what you're looking at. But I think that women's diseases in general don't get a lot of funding. And there's more allocation in certain places than other places. Sometimes it's who yells the loudest will get the dollars. There's a lot of historical pieces around that. So my fight is for the kids, and they're a protected class in research because they cannot advocate for themselves, especially when they're debilitated. So I wish I could answer your question better, but I would be interested in reading your paper when it's done. So please send it to me, okay? Thank you.
Before I call on you again, is there any other questions? Okay, right here.
No, it's okay.
How many other pharmaceutical development projects at Curemark utilize delayed release capsule technology, and is that what are your thoughts and views on that particular aspect of the application?
I think it would all depend on what type of delayed release and what we're targeting. So it would really have very much to do with what disease we're looking at, where we want the enzyme to be released. how it needs to be there, what the dosage is. It's a better question answered, actually not by me, but by my chief scientific officer, and sort of offline, which would be great. Yeah.
So it occurs to me that it's for a small company, essentially has to be a small company given the funding. Isn't it rare that you are actually trying this on other diseases before the completion of the first trial, or is that a fairly typical
Well,
pattern?
it's pretty typical to have more than one drug in development. I think that we have the intellectual property. We've done the science on a lot of things, so I don't think that's so rare. I think that we've done a lot with what is really a little bit of money, and we've raised like $70 million. So as pharma companies go, that's small, but we've done a lot with that money.
So at Athena, we've developed what we call the Athena Core 10, which is a set of kind of attributes and skill sets which we think are really key to leadership. And one of those is courage. So I wanted to ask you if you could tell us about the most courageous thing you think you've done throughout this process.
Oh, that's a good question. I think just doing it. I mean, I think just getting to a place where we went ahead and did this first trial. I think that was thinking that we can do a multi-center, double-blind, randomized, placebo-controlled study at 20 centers or 18 centers, our first one, and do it with the rigor and the regulatory expertise that we have, I think was pretty audacious. And I didn't do it alone. I had a lot of help. So I think that is something that required a lot of courage, not just on my part, but on my team's part as well.
So another one of those attributes we talk a lot about is resilience. So I want to know kind of your secret sauce on how you bounce back from what must be a number of setbacks or failures along the way.
Well, I think, first of all, I love what I do every day. So getting up and going to work every day is a great joy for me. When there's setbacks and problems, the need of the kids gets to me every time. And I think, I can't do this. How am I going to do this? How am I ever going to raise money? I'm down to nothing. How am I going to get that money? But knowing that the kids needed that, and they need it, they still need it, is what kept me going every day. And you need a lot of resilience. But and I'm a big baseball fan. People who know me, I'm a big baseball fan. What's that? The Red Sox are winning? Really?
They're discussing the 6-4 the Red Sox are winning.
Well, if it takes as long to get the 10,000th win as it did for Jeter to get his 3,000th, it's going to be a long week. But in baseball, you get up and you hit the ball, and you go to hit the ball, and you could strike out 4 times in a game, and the next day you go 3 for 4, right? So you have to know that if today's not a good day, tomorrow can be a better day. And I never know who's going to call me on the phone, or who I'm going to meet, or who's going to reach out. And so every day is like an adventure. And they're hard. They're long days. But it's knowing that we can maybe get this to the kids that really makes me get up every day and do this.
All right, so I have one last question. Any more from the audience before I ask my last one? Oh, yeah, here we go.
In the previous question, somebody had asked about the potential differences in commercialization, and in your answer, you used the word in-license, that if somebody else in-license the product. But it seems like you have a lot of personal investment in making this a success because of the drive to help these kids. So my question is, if someone did want to in-license this, if somebody approached you either about the company or the product, how would you feel about that? Would you feel that you were giving in to somebody else, or would you really want to see it through personally to make it a success?
So for me, to answer that question, the question I would have to ask myself is, who can do it better? Me or this other company? And that doesn't necessarily mean that they're bigger, they have more money, they have more marketing dollars. It just means that their focus would be my focus, and that they have the expertise to do it. So they have marketing teams and expertise that I don't have. And I'm very well aware of what I do have and what I don't have. And I mean, my team, our group. And so it's a matter of, they have to convince me that they can do it better than we can. And if so, that would be great. Because my focus is to get this to as many children who need it, and to make sure that it's priced correctly so that everyone can get it. Did I answer your question?
Which is exactly the segue to my last question, because I know many of us have been reading in the news lately about this 32-year-old entrepreneur who decided to increase the AIDS drug 5,000%. And it raises lots of issues, both in the public arena and as policy, about kind of the conflict between a drug system that's built on profit and the people it's trying to serve. Kind of, are they on a collision course? So I guess from your point of view, how do you safeguard against that, and how do we make sure that the kind of the needs of people who have health problems are really the ones that control, as opposed to the dollars that are the dollar signs that in this instance are so big that people, I'm sure, are clamoring to get a piece of it?
So I think that, that's a great question, and of course it's timely given what's happened in the news this week. But I think that, people will argue it from both sides. The drug companies will say, I did all these trials failed, I have one candidate that passed, and I have to subsidize all my other research. That's what their excuse has been in the past. That's what they've said. Now, most of them don't have R&D anymore. They get it from the biotechs or from the universities. So the model's changing, and it is on a collision course because it's a different way of developing the drugs. So they don't need to have this big R&D division that was looking at drug libraries and things like that. So I think that there's a place where companies can be profitable and not do it off the backs of sick people. At the same time, respecting that what it takes to do it, how long it takes. It's a 20 years process. And how do you recoup that? Now, there's a lot that can be said about the big organizations and sort of their, bulk, and can they streamline and do other things? But that's a bigger conversation. So I think that for us, we look at this as something that I'd like everyone to have access for, and there are companies out there that make sure everyone has access, whether that's a single copay, and regardless of their ability to pay. And so I think there are a lot of ways for us to commercialize this so that everyone gets this drug.
All right, well, I hope you all will join me in thanking Joan Fallon for an excellent, excellent Power Talk. Thank you.
Thank you very much.